this video lecture we will talk about one of the important and interesting topic from the biochemistry, that is about the glucose-alanine cycle. Now look at see here, this glucose-alanine cycle, it is related to the gluconeogenesis. Now what happened right in our skeleton muscle? They are the most busiest muscle. Now they are involved in the most of the activity of our body. Now what happened? So they use the glucose right in the skeleton muscle. So this is an glucose. Now this glucose, it is break down into 3 carbon compound, that is pyruvate. Now what happened? This process, this overall process, that is the conversion of glucose into the pyruvate, this is known as glycolysis. Now right in the skeleton muscle, the breakdown of the amino acid occur. Now this is the overall structure of the amino acid. This is the carboxylic acid group, this is the amino group, and this is the alkyl group, and this is simply the hydrogen. Now what happened when the breakdown or the metabolism, or you can say the catabolism of the amino acid occur? Now here the ammonia, it is released. Now this ammonia, it is toxic when it is present in a cell or tissue. Now this pyruvate, it is produced through the process of glycolysis. Now this is actually a keto acid. Now this ammonia, it is toxic, so this ammonia, it is converted into less toxic product. For this conversion into less toxic product or substance, this ammonia, it is transferred into the liver, where this ammonia, it is converted into the urea through the urea cycle. Now how this ammonia, it is transferred into the liver and converted into the urea, that is the less toxic product. Now this pyruvate, it will able to accept this ammonia, and this pyruvate, it is able to convert into the amino acid, that is alanine. Now this process, this is called as transamination. So transamination, it is actually the transfer of amino groups. So this amino group are the product of the amino acid metabolism, or you can say the catabolism. This amino group, it is taken by the pyruvate, and this pyruvate, the result after taking this amino group, this pyruvate, it is able to convert into the amino acid, that is alanine. Now this alanine, it is enter into the blood circulation. Through the blood circulation, this alanine, it is able to enter into the liver. So right in the liver, this amino acid is come here, the liver. Now what happened in the liver? So this process, this oral process, this is take place in the skeleton muscle, while this process, this is take place in the livers. So you can see here, this alanine, it is come into the livers. Now what happened? This alanine, it will give is amino group, and the carbon skeleton of this alanine, it is converted into the pyruvate. Now again this process, this is called as transamination. Now look at see here, this is alanine. So this amino group, it is removed from the alanine, and this ammonia, it is toxic, and it is come here into the livers. Now this amino group, through the process of urea cycle, this amino group, it is converted into the less toxic form, that is urea. Now this process occur the conversion of this ammonia into the liver through the urea cycle. Now this pyruvate, it is formed from the alanine through the process of transamination. Now this pyruvate, it is again used for the conversion of this pyruvate into the glucose. So this process, it is called as gluconeogenesis. Now this glucose, it is now further available for the skeleton muscle to produce energy. So this is overall process of the glucose-alanine cycle, which is present or occur between the liver and skeleton muscle. Now we will discuss the theory related to the glucose-alanine cycle. Now the question is that how the transamination is occur, and pyruvate is able to convert into the alanine, and again this alanine, it is able to convert into the pyruvate. Now how this process is occur? Now we will see here how the process of transamination occur. Now look at see the process of transamination, or how the pyruvate, it is able to convert into the alanine. Now look at see here, this is the structure of the pyruvate. It is actually an keto acid, and it mainly contain a 3 carbon. So it is actually a 3 carbon compounds. So you can see here, this is the overall structure of the pyruvate. Now what happened, right? This is an glutamate. It is actually an amino acid that contain an amino groups. So you can see here, this is the amino group. Now what happened? So this amino group, it is taken by the pyruvate. Now this process occur with the help of alanine transferase enzyme. Now this alanine transferase enzyme, it is able to remove this amino group, and this amino group, this is taken by the pyruvate. Now this glutamate, it is converted into the keto acid, that is alpha ketoglutarate, while the pyruvate this is able to convert into the alanine. So you can see here how the pyruvate this is converted into the alanine. So right, and so this you can see here this pyruvate it is take this amino group and convert it into the alanine, right? And this process it is mainly occur in the skeleton muscle. Now right in the liver, so this alanine it is converted back into the pyruvate, and this amino group it is released, and that is used in the urea cycle for the conversion of this amino group into the urea cycle. So this is overall the process of transamination. Now let's discuss some theory related to the glucose-alanine cycle. Now this cycle it is also known as Cahill cycle. Now why this process or this cycle it is known as Cahill cycle? Because one of the American physician and endocrinologist, that is George Cahill, because he made a significant contribution to understanding of the metabolic pathway involving amino group and carbon from the muscle. Because of this work of the George Cahill, this cycle it is gave the name of Cahill cycle. Now where the glucose-alanine cycle it is occurred? So right, this glucose-alanine cycle it is occurred between the skeletal muscle and the livers. Now next point, that is it is responsible for the gluconeogenesis. Now this gluconeogenesis it is actually the formation of glucose from the non-carbohydrates. Now right you have seen also in the glucose-alanine cycle where the alanine it is used to convert into the glucose, while we have already discussed in the Cori cycle this lactate it is also responsible for formation of the glucose. Now this alanine it is used to convert or to synthesize into the glucose. Now this is also a gluconeogenesis. While this process it is happen in the glucose alanine cycle, while the lactate it is used to convert or to synthesize glucose, this is also a gluconeogenesis. But this reactant it is converted into the glucose right in the Curie cycle that we have discussed in the previous lectures. Now what is the function of the Cahill cycle, or what are the importance of the Cahill cycle, or why the Cahill cycle it is happens? Now we have two importance of the Cahill cycle, or you can say the two important function that is performed by the glucose alanine cycle, or you can say the Cahill cycle. Now the first important that is transport ammonia to the liver to convert into non-toxic form. So you have already seen in the skeleton muscle when the breakdown or the catabolism of the amino acid occur, so it will result into the ammonia. Now this ammonia it is toxic. Now this ammonia it is converted, it is transferred into the liver, because right in the liver this ammonia it is converted into non-toxic form, that is urea. Now this conversion right in the liver, the ammonia it is converted into the urea, that is the non-toxic form of the ammonia. Though this process occur with the help of the urea cycle. So this is the first function of the glucose-alanine cycle, that is to transport ammonia to the liver, that is converted into the non-toxic form, that is urea. Now the second function, or you can say the importance, that is transport carbon skeleton to liver for the gluconeogenesis. Now when this cycle it is also responsible to transport the carbon skeleton. So you can see here right in the structure of the alanine. Now when this alanine it is transferred into the liver, so it will remove amino group. Now this amino group it is converted into the non-toxic form urea, right? The remaining skeleton or the remaining structure of the alanine it is used for formation of the glucose, that is the gluconeogenesis. So mainly overall it have two functions. The first that is to transport ammonia to the liver, where this ammonia it is converted into the urea, that is the non-toxic form. Next function that is to transport the carbon skeleton, because this carbon skeleton it is used to in the synthesis of the glucose. Now this process this is called as gluconeogenesis. So this is overall the concept of the glucose alanine cycle. Thank you so much.